Good morning, students. I am Dr. Rajib Majumder from Department of Biotechnology, Adamas University. Today, I would like to discuss on a very interesting, important topic, that is the genomic DNA library and cDNA library under Recombinant DNA Technology course. Previously, we have discussed about the different aspects of polymerase chain reaction. We have discussed in details of molecular cloning. We have discussed in details on how efficiently the nucleic acid can be sequenced through a platform like Oxford Nanopore. We have discussed in details on different kinds of the PCR compared to the conventional PCR. Now today, I am going to discuss about both genomic DNA library and cDNA library. So the point is that what is the genomic DNA library, on what is actually the library is. So library for both the genomic DNA or the cDNA is basically where we can actually keep the information of nucleic acids like both cDNA and the genomic DNA. So basically, a genomic DNA library is a collection of DNA fragments representing the entire genome of an organism. It includes both the coding as well as the non-coding region of the genome. Now, the second point is that how we can actually prepare this library, genomic DNA library. So for that, we need to isolate the genomic DNA from cells of an organism and then fragment it into the smaller piece using restriction endonucleases and also other methods. Now, basically what happens if you have a genomic DNA library, you are actually using some molecular scissors like restriction enzymes. Now the fragmented DNA is then ligated into a specific vector such as plasmid or Paj vector, and then we can actually create a recombinant DNA molecule. The insert size in this case of the genomic DNA library can vary actually, ranging from a very small fragments to the larger fragments that may span entire the genes. The library should be ideally contain a representative sample of the organism's genome, ensuring that all regions are included within that. Otherwise, we would miss some sequence of important information kept in the DNA molecule. Now, you may ask some questions that what is the application of that? Now, genomic DNA libraries are actually useful for the use and synthesis of organization of the genes and identifying the regulatory elements and also useful for genomic mapping. Now, although there are certain essentiality or importance of the genomic DNA library, but there are certain limitations for that. Genomic DNA libraries may include a large amount of non-coding DNA as well, making it more challenging to isolate the specific coding region of interest. On the other hand, a serial library is a collection of only the complementary DNA molecules synthesized from the mRNA, that is the messenger RNA, and it actually represents the expressed genes only in a specific cell type or tissue, or there are certain constitutable enzymes which are constantly available throughout the condition of a specific microorganism. And there are some enzymes called the inducible enzyme only induced upon certain conditions for the microorganism when they are induced in that condition only. So for the preparation of the cDNA library, the mRNA or the messenger RNA is isolated from the cell, and very interesting enzyme called the reverse transcriptase is used to synthesize the complementary DNA or the cDNA from the mRNA template. Then the cDNA is ligated into a suitable vector with a similar process of we do perform in the genomic libraries. So for the cDNA libraries, the cDNA library typically consists of shorter segments compared to the genomic DNA libraries, as they represent also the coding, non-coding regions of the genes at the time of isolate. So that's why the cDNA libraries provide a snapshot of the actively transcribed genes only in a particular cell type or tissue, reflecting the mRNA content only at the time of isolation. Now, suppose if we talk about the lac operon, the lac operon genes are not usually transcribed and translated throughout the life cycle of the E. coli, but when the E. coli cells uh, is actually incubated or are in the presence of the lactose only, then only these lac operon enzymes are actually transcribed and translated. বলো বলো
হ্যাঁ, বলছি ওই ডার্ক ট্যান ব্রাউন কোথাও পাচ্ছি না, যে ডার্ক ট্যান একটা জুতোর দোকানে পেলাম, সেটা হচ্ছে ব্রাশ টাইপের, বললো সব জুতোতেই করা যায়।
ঠিক আছে, মানে ব্রাউন জুতোয় করা যাবে তো?
হ্যাঁ, একটু কথা বলুন তো দাদা।
হ্যাঁ হ্যাঁ,
এটা হলো আপনার হলো ওই লিকুইড, এর সঙ্গে সেটিং করা আছে, ব্রাশও সেট করা, মানে মানে আপনি ডার্ক ট্যান কালারও টানতে পারবেন, কালো কালারে যে কালারে টানবেন সেই কালার আসবে,
মানে
ঠিক আছে?
বেসিক্যালি একটা ক্রিম টাইপের, তাই তো?
ব্রাশ ক্রিম টাইপের, ওর সাথে লিকুইড সেট করা আছে একদম। ওই টেনে দেখিয়ে দিলাম, ওনার প্যাকেজ একটা খুললাম, খুলে একটা মানে জুতোয় টেনে ওনার সামনে দেখিয়ে দিচ্ছি।
আচ্ছা, মানে ব্রাউন কালারের জুতোতে দেওয়া যাবে তো?
যে কোনো কালারে
আচ্ছা 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 আচ্ছা, ঠিক আছে ঠিক আছে, পাঠিয়ে দিন, দিয়ে দিন।
হ্যালো,
হ্যাঁ, নিয়ে নাও নিয়ে নাও।
নিয়ে নেবো?
হ্যাঁ হ্যাঁ, একদম।
কত টাকা দাম?
হ্যাঁ, নিয়ে নাও নিয়ে নাও।
ঠিক।
So, the cDNA libraries are particularly valuable for studying the gene expression. Because you know what, if we talk about the central dogma of molecular biology, even for the eukaryotes, even, so they have a lot of introns and exons, and basically only the coding regions are actually transcribed and translated to form a specific valuable proteins or enzymes. So that's why the cDNA libraries are particularly important and or valuable for studying of the gene expression, identifying the tissue-specific gene, and for cloning of full-length coding sequences. So there are certain advantages since the cDNA libraries can include only the expressed genes. They offer more focused view of the functional genes present at the particular context, whereas the cDNA libraries are commonly used in studying involving the gene expression analysis, functional genomics, and isolation of the specific genes. If you try to compare between these two, while there is some overlap between the genomic DNA and the cDNA libraries, they serve different purposes. As I mentioned, the genomic DNA libraries include the entire genome, whereas the cDNA libraries focus only on the transcribed genes. The genomic DNA libraries represent the entire genome, including both non-coding regions, while the cDNA libraries only represents the expressed genes. The genomic DNA libraries are more stable for the genomic mapping and understanding the organization of the genome, while the cDNA libraries are more valuable for the gene expressed or expression studies and the functional genomics. So here in my slide, I would like to convey the message to the students like why this is important to study the genomic DNA and cDNA libraries. The DNA library is basically the term used to describe a collection of the recombinant clones. Because in our previous videos, we have actually discussed that how to make a clones, how to make a recombinant clones, and how to actually express through through a specific transformation or mm, uh, other process called electroporation. Though there are two main types of the DNA libraries, they actually vary as I discussed. The DNA libraries are designated as being either the genome DNA library or cDNA library. Most traditional methods of library construction or preparation involve the physical cloning of various DNA fragments, if either it's a DNA of interest or a PCR that has to be cloned in a suitable vector. However, the DNA fragments are not actually clones. That's why these derived from the PCR product are becoming increasingly important in different genetic engineering experiments. So that's why, first of all, I would like to discuss on the genomic library. So cloning genomic DNA generates a population of the recombinant DNA molecules, often in a plasmid or a phage vector. Now, you know what? As in my class also, I have mentioned that there is different kinds of the cloning vectors. But you know what? This is very important to understand that at what condition, which kind of the vector one should use. So being a recombinant DNA technologist, you have to have an idea, clear-cut idea that what is the length of the DNA of interest that you want to clone, and for that, what kind of the vectors that you have to use for your purpose. It may be a bacterial cell, or it can be a phage particles, or it may be a BAC or YAC like this. A collection of independent clones is termed as a clone bank or library. For example, suppose if you have your Adamas University library where there is a combination or cataloging of the different kinds of the books. So from that, For example, if you want to study the enzymology, so there are several kinds of the book. One you can pick at a time to study the enzymes or enzymology. So genomic libraries are used to find the genes, and we can compare the genes from the different organisms. The gene libraries are made when the entire DNA is from a particular organism and digested into a specific fragment using restriction endonucleases. And each of the fragments is then cloned into a suitable vector and transformed into an appropriate host. A genomic library contains DNA fragments that will actually represent the entire genome. A genomic library should represent the entire genome of an organism and as a set of overlapping clone fragments, while will actually therefore enable the isolation of any sequence of the genome for our experimental purpose. So it is important that to maintain a genomic DNA library, and the cloning should be ideally generated by the sequence independent procedure, ensuring that the fragments are only generated at a random and thus there is no bias towards a particular sequence of the interest already present in the reaction mixture. And finally, the clone fragment should be maintained in a stable form while no misinterpretation of the sequence due to the recombination of the different replication of other clone DNA during the propagation of the recombination. While these criteria may seem rather demanding, the system available for the cloning genomic libraries enable them to for the requirement and they met more or less completely. So there are different steps of genomic library preparation. First, isolation of the genomic DNA from an organism, for example, E. coli, algae, fungi, yeast, human being. Digest the DNA with different restriction enzyme, linearizing the suitable cloning vector with the same restriction enzyme so that they should have a same type of overhang region for prop or better ligation process. Mix this cut chromosomal fragment with the linearized vector, and we have to ligate using some ligation of ligase enzyme. And then after ligation, when there is a recombination, we need to transform or through heat shock method or electroporation into a suitable host. And finally, we need to isolate the large number of the transformant E. coli from the host. So it's a specific way. So restriction enzyme we are cut. You can see the image that we are entire the genomic DNA. We are actually cutting through the restriction enzymes. We actually get to see the different fragments of the DNA collecting of the overlapping regions. And finally, we need to actually find out the non-overlapping fragments. And finally, we need to actually insert the DNA into a suitable vector. And finally, we need to clone for proper screening. Now, you know what? The restriction endonucleases have an influence on the library. The restriction sites are not evenly spread in the genome. Some insert will be large and some insert will be very small. A 4-base cutter will give a mixture of mostly small fragments, whereas a restriction enzyme that has a 6 and 8, even bigger than that recognition site, will generate some larger fragments. সো বাংলায় বললে যদি স্মল সাইজের হয় তাহলে স্মল ফ্র্যাগমেন্ট ক্রিয়েট করবে, যদি লার্জার সাইজ হয় তাহলে লার্জ ফ্র্যাগমেন্ট ক্রিয়েট করবে। Even if an enzyme can recognize a 4-base pair recognition sequence is used to digest the entire genome, so there may still the segments that are too large or to be cloned. To avoid this, partial digestion is often used. The enzyme is allowed to cut the DNA and only in the short time, and many of the representative enzyme sites are not cut, leaving the larger piece of the DNA fragments. In addition to that, it is useful to construct another library using the different restriction enzyme. So the first consideration of the constructing the genomic library is the number of the clones required. So there are important formula by which we can actually find out the different number of the clones as here. The size of the genome will actually determine the number of the clones. A small genome such that of the E. coli will require the fewer clones than a more complex organism such as the human genome. Obviously, যার সাইজ ছোট হবে তার কমপ্লেক্সিটি কম হবে, যার সাইজ বড় হবে তার কমপ্লেক্সিটি বেশি হবে। The type of the vector to be used will determine the size of the fragments. Now, for MSc students, uh, in your present screen, you can find this important formula. And you know what? In any important competitive examination like GATE or NET, especially, you may find at least one numerical while you have to use this formula to get a 4 full marks in the NET examination. While it's basically n is equal to ln 1 minus p divided by ln 1 minus a by b. Well, the capital N is the number of the clones required when we try to clone the genomic library. P is basically the desired probability of a particular sequence being represented. So we actually typically set it to have a statistical significance 0.95 to 0.99. Small a is the average size of the DNA fragment that we want to clone, and b is the size of the total genome expressed in the same as units same as e. So we can actually use this formula to find out the answer of the typical question. So it's a problem number one. You can actually do in by your own using that same formula. That a specific lambda EMBL3 is used as a vector to clone 20,000 base pair of the fragment generated from a partial digestion using a specific restriction endonuclease of the human genome having a size of 3 into 10 to the power 9 base pair. It's a huge. So then we used to actually isolate the gene containing completely on 20,000 base pair of the fragment. That's why as a probability we have a 99% of the chance of isolating the DNA in the lambda EMBL3 insert of the recombinant genomic library. So the question is that how many independent clones we must examine. So you can use the formula that I have already shared in the previous slide, this formula. And using the formula, you can find out the answer of the question. So the cDNA library, a library is typically made up from the mRNA that has been reverse transcribed into by the enzyme called reverse transcriptase. The reverse transcriptase produces the complementary DNA fragment which are then cloned into a suitable vector. Therefore, unlike the genomic DNA library, our cDNA library will actually we use for only the expressed genes in a particular cell type at a particular condition of the cell being reproduced. So I've already discussed that how to construct the cDNA library, their usefulness, and it's a basic comparison between the cDNA library that in the library, cDNA library, we need to extract the mRNA, then cDNA synthesis using the reverse transcription, and then insert this cDNA library into a suitable vector to make it a recombinant. And finally, we can transform it to suitable vector, I mean suitable host. On the other hand, in the genomic DNA library, we need to extract entire the genomic DNA. Then we have to partially digest it through restriction enzyme. Each and every digest is then fragmented and cloned into a suitable vector to make it a recombinant. And finally, each and every fragment, recombinant fragment should be transformed into a suitable host. So for the cDNA library clones, another interesting numerical you may find using the formula. And I must tell you that please uh, practice this numerical, because in any competitive examination you will get at least one numerical of question of 4 marks. And it's a direct numerical that just you put the values in the numerical in the formula, and you will get the answer, and you will get 4 marks. And if you get a 4 marks, obviously your chances of selection within the GRF for the LS will actually increase substantially. So it's n is equal to ln1 minus p by ln1 minus n by t, while capital N is the number of the cDNA clones, capital P is the probability, while we set it 0.99 or zero point 0.9, that is 99% chance of identifying the mRNA representing the cDNA. Small n is the number of the molecules, that is the mRNA present in the cell, and capital T is the total number of the mRNA molecule present in the cell during that condition of the incubation. So it's the second problem. So I have actually discussed two problems using two different formula. If you practice these two, I'm sure that you will get full marks in any competitive examination, because you will get at least one out of these two problems, either to finding out the number of the cDNA clones or number of the genomic DNA clones using any of these two formula, and you will get a full marks for that. So for that, with this note, I'm concluding the session and hope you have enjoyed the session. And whenever we will be in the class, uh, you just work out the problem, and I will actually see from your copy that whether you have successfully executed the formula or the numerical, and then I will discuss in details also in my class. Okay, thank you very much, student.